Hi guys, welcome to the third lesson of high school biology. Today I'll be talking about the overview of cell respiration. So since we only have 30 minutes, it's kind of gonna be like impossible to talk through all the details of like glycolysis, Krebs cycle, electron transport chain, and fermentation and more. So instead of giving you guys very specific details on each of the cycle, I will be talking about it very briefly so that we can all understand the overall steps of cell respiration. So first, let's talk about what goes in and out during cellular respiration. First, the equation for cellular respiration can be represented through this. So I'm going to write equation. The equation is going to have glucose, which is C6H12O6, plus oxygen, 6 of oxygen, which will produce carbon dioxide, CO2, 6 CO2. Actually, let me just write that again. 6 CO2. And water, H2O, and ATP. ATP. Or we could call ATP adenosine triphosphate. To simplify the chemistry terms, this is basically sugar added with oxygen or air, which will then equal carbon dioxide. And water. And ATP is basically energy. It is important to note that this equation is for an aerobic respiration. So aerobic respiration. And there is a separate step for anaerobic respiration, which we will be covering today as well. So first, let's talk about the long, scary word adenosine triphosphate, or ATP. It's made up of adenosine and 3 phosphates, hence the name adenosine triphosphate. So you can write consists of adenosine and 3 phosphate, which then equals adenosine triphosphate. So ATP is a molecule that stores energy for immediate use within the cell. Basically, it provides energy to the body when the 3rd phosphate breaks off from the rest of the molecule. This produces another molecule called adenosine diphosphate, or ADP. So ATP, so how energy works in ATP is that we break off one of the phosphates. Remember how we said it was triphosphate? We break off one of the phosphate, and that produces the energy. And what will end up is that ATP will now become ADP, adenosine diphosphate, equals So ATP is very important since the whole point of cellular respiration is to gain this So, moving on to mitochondria. So many cycles of cellular respiration takes place in the organelle called mitochondria. Mitochondria was mentioned before in the previous video where I explained their functions, but this is basically the powerhouse of the cell. So mitochondria is enclosed by two membranes, the outer membrane and the inner cristae membrane that is folded. So you can write that. It is enclosed by two membranes. And as we mentioned before, that consists of outer membrane and inner membrane. Inner cristae membrane, sorry. So the inner membrane, inner cristae membrane, divides the mitochondria into two internal compartments, the outer compartment and the matrix. So you can separate this by outer compartment, and the matrix. If you look at the figure right here, you can see that the matrix is right over here in the mitochondria, and the outer compartment or the outer membrane is surrounded by here like this. The matrix is actually really important because this is where the Krebs cycle takes place. And now we know where the structures and the ATP and everything. Now we're going to move into the actual steps of cell respiration. So I'm gonna briefly describe the steps. So if oxygen is not present or if it's anaerobic, a process called glycolysis is followed by alcohol fermentation or lactic fermentation. But if oxygen is present or if it is aerobic, glycolysis is going to be followed by the Krebs cycle, the electron transport chain, and chemiosmosis. So glycolysis is going to actually happen in both aerobic and anaerobic process. So we can see that in both of them that they share the same process called glycolysis. So in this process, it doesn't matter whether the cell has oxygen or not, because it can work without the O2. So it's very important to note that glycolysis does not require oxygen. We can actually understand what is going to happen in this step by understanding the word glycolysis. So let me just write the word glycolysis here. Glycolysis. So the word glycol sounds really similar to the C6H12O6 molecule that we were talking about earlier, which is glucose. So glycolysis as glucose. So glucose. And the lysis actually means to lyse something or cut or break down something. So we can write means to lysing. So if we put these two words together, we have lysing glucose or breaking down glucose. Say. And this is exactly what glycolysis does. Like, it basically breaks down glucose in this step. And the step occurs in the cytoplasm. In the cytoplasm. So it's important to mention that glucose is a 6-carbon molecule. So glucose is a 6-carbon molecule, which means it contains 6 carbons, basically, like this. And in this step, remember, we're breaking down glucose. So with the help of 2 ATP to supply the energy of activation, glycolysis happens by breaking glucose into 2, producing a new molecule called pyruvate, or pyruvic acid. So it's going to break down to 2, called pyruvate. And pyruvate. 1, 2, 3. This new molecule, as you can see, is going to contain 3 carbon molecules each. So if you add these 2 pyruvates together, we get 6 carbon molecules once again. So it kind of makes sense. The glucose is going to be broken down into 2 pyruvates, which both have 3 carbons. So if you add these 2 together, then it's going to equal glucose again. Okay, so now we got that. So overall in this step, glycolysis is going to release 4 ATP molecules, resulting in a net gain of 2 ATP. So you can write that it is going to gain 4 ATPs. ATPs with a net gain of 2 ATPs. Why a net gain of 2 ATP? Because remember before we used 2 ATPs, the other 2 ATPs to activate as like a source of energy of activation. So what gets the cycle going. So that's why we end up with a profit basically of 2 ATPs. So to sum up what we have gained through this process, we used 1 glucose and 2 ATPs to produce a net gain of 2 ATPs and 2 pyruvates and 2 NADHs. We're going to talk more about the NADH, which you guys are I'm pretty sure are unfamiliar with, but we're going to talk about this molecule more in the next step called the Krebs cycle. So this step is the first step that requires oxygen. So if the cell does not have oxygen, it will not complete the step. So it's important to note that this is going to be the first step or the first stage of aerobic phase of cellular respiration. So this cycle is called the citric acid cycle or the Krebs cycle. They're both used interchangeably, so don't get confused by that. And remember the pyruvate that we produce from glycolysis? Well, this is going to combine with the coenzyme A to form a new molecule called acetyl-CoA, which will then enter the citric acid cycle. So if we can kind of summarize what we have done before in glycolysis, we used glucose to be separated into 2 pyruvates, pyruvate, and then pyruvate. And then now, the pyruvate is going to be combined with the coenzyme A to form a new molecule called acetyl-CoA. And now, this is the preparation step right here, where it basically forms acetyl-CoA. That is the preparation step. Now we actually get into the Krebs cycle. So, as mentioned before, Krebs cycle will occur in the matrix of mitochondria. And to start off the process of citric acid cycle, the acetyl-CoA is going to combine with a compound called oxaloacetic acid. Oxaloacetic acid, yes, it's kind of hard to say, but as acetyl-CoA goes into the cycle, there is another molecule or a compound called oxaloacetic acid. And when these two molecules and compounds are added together, they form something called citric acid, hence the name citric acid cycle. Citric acid. And now the citric acid is going to be oxidized through the Krebs cycle to produce many molecules actually. Like this, it's going to go around in a cycle. Oh, that's an ugly circle, but it's okay, we'll have to deal with it. So within this cycle, the citric acid is going to get oxidized, and it's actually going to have the NAD plus form into NADH. And this will happen 3 times along the cycle. So let's put another NAD plus here with NADH here. And let's put another one here, since we're running out of space. NADH. It is also going to form ATPs. So an ADP is going to be formed into ATP during this cycle. And of course we have the last one is called FADH2. So FAD is going to be transformed into or oxidized into FADH2. So yes, so each turn of the Krebs cycle produces 3 NADH. Here, I'll label it. It produces 3 NADHs, 1 ATP, and 1 FADH2. But remember, in glycolysis, we separated the glucose into 2 pyruvates, right? So basically, this whole process happens in this process. It's in here too. So we have to twice the amount of what we had gained from this one cycle. So in reality, we will have 6 NADHs, 2 ATPs, and 2 FADH2s. And the FADH2 and the NADH is really important since it is going to be the output in our next cycle called the electron transport chain. So since the electron transport chain is such a detailed process, I'm going to tell you guys the overall function of the ETC. So the FADH2 and the NADH we produced from the previous cycle transports highly energized electrons from the citric acid cycle to the electron transport chain. And the ETC, or the electron transport chain, actually takes place in the cristae membranes of mitochondria, as we talked about it before. And so the energy from the energized electrons being pulled through the ETC powers the pumping of the protons across the cristae membrane from the inner matrix to the outer compartment against a gradient. So if you see right here, this is the outer membrane over here, and what ETCs wants to do is basically create a proton. 어? 진짜? 어떡해?
괜찮대. 그냥 하면 될 것 같아.
Okay.
중간부터.
Okay.
소리가 왜 그래?
Oh.
오디오가 여기서 나가면 좋을 것 같아.
Wait, maybe it's because I undid this before?
거기서부터 다시 하면 돼.
Okay. So the whole purpose of ETCs are to establish a proton gradient that's and
So
that's
the
all
whole
you ever
purpose of ETCs are to
that's all you ever need to know about the functions of ETC. So we can write that in our slide. Is to establish a proton gradient. Then the proton gradient established by the electron transport chain in mitochondria is going to represent an enormous potential energy to power the phosphorylation of ADP to ATP. So in the outer compartment of mitochondria, when protons flow down a gradient through ATP synthase channels, their potential energy is transferred to kinetic energy and then to chemical bond energy stored in the molecules of ATP. And again, the whole purpose of cellular respiration is to produce ATP, and this is how we're going to get them. And the production of ATP by this step is called chemiosmosis, or otherwise known as the oxidative phosphorylation, and this produces the most ATP in the aerobic cellular respiration. So now that completes the overall aerobic cellular respiration, and now we can move on to the anaerobic cellular respiration, where in this case the cell does not have oxygen. So when oxygen is not present, we have to go through an anaerobic respiration, or so-called fermentation. And fermentation consists of our previous step called glycolysis. So if we can write here, from glucose we start with glycolysis, and then we start with glycolysis, and then it's gonna go to the pyruvic acid, and then from there it goes on to either alcohol fermentation or lactic acid fermentation. So So let's talk more in depth about the alcohol fermentation. So alcohol fermentation is a process by which certain cells convert the pyruvate from glycolysis into a ethyl alcohol and carbon dioxide. So it's going to transform the pyruvic acid into this thing called ethyl alcohol and also produce carbon dioxide as well. So in this fermentation, the fermentation is mostly used by the baking industry where they produce carbon dioxide,